It's my pleasure to introduce Jeff Rathmell, who's today's seminar speaker. So Jeff carried out his PhD with Chris Goodnow at Stanford University, where he worked on the role of FAST in enforcing immune tolerance. He then moved for his postdoc to the University of Chicago with Craig Thompson, where he continued to work on cell death, this time working on molecules involved in apoptosis. So, for example, he published a landmark paper about the role of BAC and BACs in thymocyte development with Craig, and also began his interest there in the role of metabolism. He then moved to Duke University, where he started as an assistant professor and rose through the ranks to become full professor there, with a major focus on the role of metabolic pathways in T cells. More recently, he's moved to Vanderbilt University, where he is a professor and director of the Center for Immunobiology, and he continues to be interested in immune metabolism. And the talk of his title today is Fueling T Cells in Inflammation and Cancer. So I also want to remind you that later this afternoon at 2:30 on the second floor of WMBB, there's going to be tea time if you want to meet and talk to Jeff some more. All right, so please welcome Jeff.
All right, well, thank you, and thanks for the invitation to come. When you're coming from the south, it always seems like the worst possible idea to come to Minnesota in January. But I guess this is relatively not as bad as it could be. I was talking to Steve. At least there's no active shooters in my hotel, so that's good. Okay, so thanks, Mike, for the opportunity to come here and introduction. So I've been at Vanderbilt for 2.5 years, and was at Duke for about 12 and a half or so before that, where we've been interested in immune metabolism and apoptosis pathways for the most part, and it's sort of shifted to metabolism and to sort of cell fate a little more broadly. So before I start, though, so this is I'm not sure if about the same. So this is the village of Rathmell. So I just point this out. So Rathmell is a little town in North Yorkshire, England. You're practically up to Scotland when you're up there. And it is right way between Giggleswick and Wigglesworth, but it's a beautiful little area. You can see the trees down there. So I encourage you all to visit. So anyway, with that, we'll begin. Okay. There you go. So just a couple disclosures. I would just point this out mainly because I'm going to talk about using a compound from this company, Calathera, that's provided us a little funding. Okay, so again, we've been interested in metabolism and immune responses, and of course this is a generic cartoon of the immune response, but so you sort of go through it from a metabolic perspective. So resting immune cells, and we're going to talk about T cells. B cells are very similar, but focus on T cells today. Resting T cells, of course, they're in more of a surveillance mode, so they're moving around. So if you think what do T cells do when they move around, that needs ATP. It's a catabolic metabolic program, so they have to be generating, burning things to generate energy. A lot of that's lipid sort of oriented, but they're just moving around. And once they encounter a pathogen or a neoantigen or a self-antigen, whatever stimulates them, of course, then they are encouraged to rapidly expand. So they have to switch from burning fuel to generate energy to now building. So now you need to be able to make RNA and DNA and protein and lipids. So now it's all anabolic. So there's a whole catabolic to anabolic switch that happens in this growth meiosphere, in this expansion. And of course, once the cells are growing, they're not just proliferating, they're also differentiating. They're gaining different effector fates. There's a lot of plasticity and a lot of different fates there. But they gain their specific roles over the course of that. And so that's why I have the multiple colors here. And of course, at the end of the response, you have to shut it all back down. It was contraction mode where the cells are tuning back down from anabolic back to catabolic so they can re-enter sort of this survival and surveillance mode here as memory cells. So there's this whole cell switch in terms of what the cells are doing. And it can be sort of overly simplified, and I'm as much of a culprit of this as anybody, in that we just sort of think about glycolysis, but aerobic glycolysis is really the process that changes the most in the course of this. So aerobic glycolysis just means glycolysis in the presence of oxygen. It doesn't mean that they're not doing other things. It just means it's glycolysis in the presence of oxygen. So glycolysis increases a lot, so you can almost sort of replace this axis here with the extent of glycolysis. But like I said, it's not just glycolysis, and a lot of things change. And so we've done a lot of work on trying to understand the different pathways that change and how the roles that they play. And so this is just one illustration of this. This is a semi-targeted metabolomics experiment. We take naive T cells or T cells that are stimulated for 1 or 2 days, and you just look at the metabolome. And what you see is the naive T cells, they have a certain profile here, but after 24 hours and 48 hours, it all switches. And so you see there's a global switch. It's not just that glycolytic intermediates go up. A lot of things change. And most of these you can pull out and identify them, and they're in all sorts of anabolic pathways. Again, just fits this idea T cells are turning everything on. Right, so they're turning up all these pathways. Now, this aerobic glycolysis and this kind of switch is best described and best known in cancer settings. But it's interesting, if you take this sort of these proliferating T cells out here 48 hours, and you compare them to leukemic T cells. Isn't that there? Oh, okay, it didn't come up. We'll see if I can actually. No, I got a little thing. Oh, it's not there. Anyway, it's weird. It's on the screen, but it's not here. Okay, I'll have to look at the screen. All right, so this is a T cell leukemia, TALL. So it's primary T cells as well, and they're also doing aerobic glycolysis by sort of your standard measure, but their metabolic program and profile is entirely different. So the TALLs and the activated leukemic cells and activated T cells they don't necessarily have the same program. So that just makes the point that it's a lot more heterogeneous in terms of the actual pathways that cells use. So that was all in vitro. So this, of course, all happens in vivo. And just for time, I'll skip over this really quickly, other than to say if you use PET imaging, which measures glucose uptake, of course, this is used a lot in cancer. And so cancer cells take up glucose, but you sort of pick your favorite immune disease, and it happens in there as well. So these sort of metabolic changes are widespread in vivo. Okay, so that's activation. When I mentioned, of course, the cells differentiate. They have different functions, and they also have different metabolic profiles. And, of course, there's lots of different variations on this and plasticity, but just to keep it simple, if we think about Th1s, 2s, 17s, and Tregs, the different cues to differentiate them, and they all have different metabolomes. Again, it uses the same sort of non-targeted or semi-targeted metabolomics approach, naive T cells, Tregs, Th1s, Th17s. So they're all metabolically distinct. Okay, so you can make some generalizations from this. One is, again, they're all different, but the effector cells, the Th1s, 2s, 17s in this case, they tend to have higher rates of glycolysis. Tregs have lower rates of glycolysis, but they're more mitochondrially based. Now, none of this is all or none. The effector cells are still doing mitochondrial metabolism. Tregs are still doing glycolysis. It's just sort of overall preferences. Right, so there's quite a bit of heterogeneity. So one of the questions that we've had in our lab is, how does this develop, and what does this mean for the cells? And so what I showed you before was pretty straightforward in like 5 days after the stimulation. So we've gone back and done this with a few different conditions to try to better understand how these different metabolic programs sort of evolve. And so if you take a number of different conditions here, a few different sort of Th0, Th1 conditions, a couple different Treg, a couple different Th17 conditions, and just try to see how does their metabolome change over time. And so even within 24 hours, and this is a time point where it's kind of hard for us to distinguish them at a transcription factor level. So they're not really, they're starting to differentiate, but they're not very far down the pathway. But you can already segregate them based on their metabolomes here. And this is just the top 100 or so metabolites, where the Th1s, the Tregs, and the Th17s are all kind of segregating very early on. So these metabolic programs start to develop pretty early. And they just get more and more solidified and more specific as you go. So by 72 hours, you can get a different program for each of these different cytokine combinations, to the point where you can almost pick out what the cell got based on the metabolome of the cell. And one of the things that's nice about this is you can take these kind of data and pull out specific signatures or metabolic profiles. And some of these signatures, again, a lot of this is catabolic, anabolic sort of metabolism, such as growth. So some of this just has to do with the rate of cell proliferation. And so some of the metabolites profiles fit the slow cells and some fit the fast cells. But others are a little more specific. Like there's a TGF-beta signature, for example. Regardless of how fast the cells are proliferating or what differentiation path they're taking, TGF-beta gives you just a certain metabolic signature. And likewise, you can pull out signatures that are specific for each of these other sort of fates here as well. So these kind of signatures give us the idea that these metabolomes really do fit the fate of the cell, and now we want to understand why. Okay, so one of the things you can do from these sort of data is try to pull it out and try to see what are the things that are really unique to a given subset, and so you can build these metabolic networks. So this is a network that we did comparing Tregs and Th17s, and I know you can't read it, but just the point to make here is there's a few things that really stand out. So nucleotide metabolism is very different between Th17s and Tregs. Serine and 1-carbon metabolism stands out, as well as glutamine metabolism. And so from these sorts of things, we're now trying to pull out specific new targets that might be sort of specific metabolic dependencies of one subset versus another, or important for the differentiation or function of one or another. Right? And so I want to use that as kind of a way to launch into two questions to talk about today. One is, what is the metabolic phenotype of a tumor-infiltrating cell? And that's kind of a specific question, but the reason for it is everything I showed you up to this point is there's all these different metabolic phenotypes, but that was all in the context of a cell getting an inflammatory cue, but having every nutrient it could possibly want. But you go into a tumor, it's very different. Now all of a sudden the glucose is different, hypoxia, and so the cells have to adapt to a sort of a hostile environment. And then the next question is, what are the nutrients that are really essential for T effector cell function? If they have to have these profiles, what are the ones that are really important? Because those are the ones you want to be able to target or understand. All right, so let's start with talking about the tumor cell. So I mentioned that the tumor microenvironment is sort of metabolically hostile, is sort of a good way to think about it. So again, everything I showed you up to the point is in adequate nutrients, everything's there that the T cell could possibly want. But of course, in a tumor, that's not the case. And so this is, on the left, these are images from a mouse xenograft, where the xenograft, so it's not particularly physiologic, but nonetheless, it sort of makes a point. If you take the tumor out and you can sort of enzymatically image the levels of glucose and lactate in this case, you can see in these tumors there's regions where the glucose levels can be largely depleted. So in this case, it's not detectable, so it's down to 0 in their measurements. Whereas you get other areas where the glucose levels are fine. And lactate is just the opposite. We have levels where lactate is typically just sort of low millimolar range. But then you can get areas where lactate can be up to 40 millimolar, okay, which is about 20 times higher than it should ever be in sort of a normal scenario. So this is very, this is not a good environment. If you have a T cell that needs glucose, this is not a good place for it. And of course, this is just glucose and lactate. You have to think of all the other metabolites and nutrients that are changing. And oftentimes we think about hypoxia, but everything could be changing. Okay? And then there's lots of other things too. So inhibitory signals that are also affecting the metabolism of the T cells. PD-1 and CTLA-4, of course, these are the big checkpoint regulators. There's a lot of attention around these in sort of the immunotherapy space. But they are quite potent metabolic regulators for T cells as well, just directly. Through signaling, through modulating PI3 pathways. And then there's all sorts of other things that can also affect the T cell metabolism. So it's a tough place for a T cell to work. So we wanted to know what do actually T cells look like in tumors. So the place that we began was we went to the tumor genome, or the TCGA, Cancer Genome Atlas, and tried to see, well, what is the tumor, the solid tumor, that has the most sort of T cells routinely? And this would be separating out any lymphoid or liquid tumor, sort of just setting them aside and just focusing on solid tumors. And if you do this and just line them all up based on CD8 expression in the TCGA, the one that's on the far right is clear cell renal cell carcinoma, or CCRCC. So it's there. It routinely has the highest CD8 T cell signature. It has a lot of CD4s as well, but the CD8s are what really sort of stand out. And we get these tumors into the lab. So patients come in. The first thing that happens with these patients is the tumor is resected. And so these are treatment-naive patients, and then we can look at the T cells in the tumors. So this is from one of the patients, just to stain for CD8 cells and just show that yes, there's a lot of CD8 cells in these tumors. Okay, so now T cells in tumors are sort of known to be non-functional, and so that's what this slide basically shows. So if we take the kidney tumors and either healthy donor peripheral blood or peripheral blood from the same patient and look at the function of the T cells. On the left, this is some activation markers with some healthy donor T cells, just to show that we stimulate them. The TILs don't stimulate as well based on CD25 or CD71, or even just blasting for cell growth here. Proliferation on the right, control T cells, when they are stimulated, they divide as indicated by diluting this dye, cell trace violet, each peak representing a subsequent division. Peripheral blood from the patients will do this pretty well, but the TILs, not so much. There's a few cells that'll proliferate, but for the most part they don't. So T cells from tumors are defective, right? That's kind of known. All right, so what is the metabolism like of these cells? So what we do is we take the tumors out, we make a single cell suspension, and just culture it. Now they have full media, so now in principle they have every nutrient that they could want. We just want to see what are they doing and how have they adapted to the tumor microenvironment. And so one of the things that's a little surprising is even though they have glucose now in the culture media, they don't take up a lot of glucose. They still just don't use it. And so using this fluorescent glucose analog here, 2-NBDG, so you control T cells when they're stimulated, they start to take up the dye, the fluorescence shifts to the right. TILs don't really do that. They don't turn it up, and it's quantified on the right. So they've turned down their glucose metabolism, would be one way to infer this. And the reason we think they've done this is actually they express a good amount of the glucose transporter, GLUT1, so that's not doesn't appear to be a defect. But what we think is the problem is they don't actually transport they don't move the transporter to the cell surface. It doesn't traffic to the cell surface. And one way to sort of indicate that is phosphorylation of GLUT1 on the serine 226 site. And the shift is pretty subtle here. It's not a very good antibody in reality. But this site has been shown to regulate the trafficking of the transporter. And ordinarily, that phosphorylation increases at least a little bit here. It doesn't really happen in the TILs. So we think the TILs have a problem with transport. So this is a picture of what an anabolic cell should do. And this actually comes from one of Craig Thompson's reviews here for talking about cancer. But T cells should be the same. But there's a defect in glucose uptake. Now, it's not just a defect in glucose uptake in these tumor-associated lymphocytes. If you go down the glycolytic pathway, there's a few other problems. And one that's worth mentioning is actually GAPDH. So this is, of course, the enzyme that is often used as a loading control. But it's actually downregulated pretty significantly in these TILs. So GAPDH, of course, is right in the middle of the glycolytic pathway, sort of at this big juncture point in the pathway. But GAPDH is downregulated. So they have two defects in glycolysis. So glucose metabolism upstream has some adaptations and some defects. So what about the mitochondria? That's the next step where it kind of integrates a lot of this. So we looked at mitochondria, and I'm just going to show just a tiny bit of data on this. So we started this by just imaging the mitochondria, see what do they look like. So these are confocal images, Z-Stack series going down mitochondria for control T cells or TILs. And what you can see in the control T cells is mitochondria, they tend to be sort of aggregated and connected into these networks, so sort of a fused network. Whereas in the TILs, you get some of those networks for sure, but there's a lot of disaggregated, small, fragmented mitochondria that are spread around. So the mitochondria is sort of a different morphology. And you can look at by EM to get I guess, a cleaner view of this. And just zero in on the high-resolution pictures of the zoom-ups here. The mitochondria from the healthy T cells, they look big and fat and round like they're supposed to. But the mitochondria from the TILs are all, again, they're small, they're fragmented. They're in a configuration that you'd call condensed. The cristae are sort of extended. So this is a structural configuration that tends to associate with cells that are not very efficient at respiration. Okay. So you quantify this, and you can see the control T cells have quite a distribution, but the TILs really favoring these small, round, fragmented mitochondria. Okay. So this has functional implications. I can mention the respiration defects. But if you look at the mitochondrial membrane potential, TMRE here, again, it's typically kind of a mix, but we have this enrichment for T cells with hyperpolarized mitochondria here. Now, hyperpolarized mitochondria, one of the things that will do is that can lead to increased generation of reactive oxygen species. And so if we look at that, we see the same thing. So mitochondrial reactive oxygen species are pretty significantly increased in the TILs. Of course, this can be a bad thing, but cells have ways to adapt to neutralize that. And so actually, conversely, this total cellular reactive oxygen species stress, just using a dye like DCFDA, which is kind of a standard way to measure this, that's actually reduced in the TILs. And we think the reason for that is these T cells have adapted. They're taking up whopping amounts of cysteine and have very high glutathione levels. So we think they've turned on antioxidant processes. All right, and we think that's the cause of that. All right, so then they have this sort of phenotype here where there's these defects in glycolysis and defects in mitochondria, fragmented mitochondria, ROS, and there's some adaptations to that. So that's all descriptive of what the T cells are like. The next question is, what does that metabolism actually matter to the T cell? We've done a few things to test, can you modify the metabolism and have an effect on the T cell function? And we're just going to show you one, and that's if we just give pyruvate back to the cell. Now, the reason to give pyruvate is it can go into the cell through the same transporters that would normally excrete lactate. But once pyruvate's in the cell, of course, you bypass any defects in glycolysis. You provide a pretty potent fuel for mitochondria, and it's also a good reducing agent. So it kind of hits three of the problems in one shot. And if you do this and then activate the T cells, what you find is that these are all TILs here now, but there's the resting stimulated or stimulated with the addition of pyruvate to the media. They make more granzyme B, they make more interferon gamma, they make more TNF. So the T cells are more functional by giving them this additional metabolic fuel. fuel. Now, of course, pyruvate is nothing that you're going to use therapeutically. But the reason I think this is actually kind of an important experiment, because it makes the point that the metabolic changes and adaptations in the T cells are actually directly contributing to the functional defects that the T cells have. So these metabolic changes matter. Right? So that's the bullet point here. These metabolic defects directly contribute to the functional defects. So understanding them will hopefully direct us in some new ways. So that's about all I want to say about the TILs. I want to switch to the other project now, because all that says is, okay, the metabolism matters, is sort of what that says. So if it matters, what do they really need? What are the minimal components? Right. So there's a lot of different fuels that T cells can use. They can fall into three broad categories: glucose, of course, glutamine, and then lipids. I'm not really talking about lipids. I talked about glucose already. I'm going to focus more on glutamine. But we've done a little bit on glucose just to make this point with one figure. And that's if we knock out the glucose transporter GLUT1 and do an inflammatory bowel disease model here. It's a T cell transfer model. Normal T cells can induce IBD. Of course, this is very well described. You might start to lose weight. But if the T cells don't have the glucose transporter GLUT1, they really don't cause disease. So the T cells have to be able to take up a lot of glucose to be able to induce this inflammatory setting. So glucose, got a little check mark up there. All right, so what about glutamine? So again, glutamine is another fuel that has been well described, mainly in the cancer metabolism field, to help support and go in concert with glucose. So it kind of makes sense that glutamine should be important. And glutamine is actually used for a lot of things. Now, I'll get into that a little bit more. But the first experiment is just to take the glutamine away from the cells. Now, when you culture cells, oftentimes you add extra glutamine to the media. But you can also do the experiment where you just take the glutamine away and see what happens. And so this is if we take the glutamine away from T cells that are differentiated to Th1s or Th17s. And what you see is the T cells don't proliferate as well. They don't differentiate as well. They don't do anything as well. So they need glutamine to a certain extent. Now, they don't need it absolutely, because you can see some proliferation here, as indicated by diluting the cell trace violet. And actually, the Th1s are doing slightly better than the Th17s. But glutamine does have a pretty significant role. And this Th1, Th17 difference, I'm going to come back to that a lot. All right. So glutamine has a role. So what does glutamine actually do? So glutamine comes into cells, and it's actually a very simple metabolic pathway. It's only just a couple steps. So glutamine gets converted to glutamate by glutaminase. Glutamine synthetase can send it backwards. Glutamate dehydrogenase will convert it to alpha ketoglutarate. Now, these have lots of different roles. Glutamine is a nitrogen donor for nucleotide synthesis. Glutamate also does that same thing, and it regulates redox balance. And then there's this very important switch with aspartate here through the glutamate transaminase. Alpha ketoglutarate, of course, this is a metabolite in the TCA cycle, so it's a central mitochondrial fuel. But it's also got a sort of more and more apparent that it's got this big role in regulating epigenetics through demethylation. Okay, so this pathway is linked into nucleotide synthesis, redox balance, central energetic sources, as well as epigenetics. Right, through these three metabolites. Right, so what does it really do? So we've gone to our metabolomics data and trying to see, well, how much does this change? So this is just pulling out a few of the metabolites. You have glucose coming in to give you pyruvate, and then this is the TCA cycle here, and glutamine feeding in, at least the main relevant metabolites. If we take T cells either naive or stimulated with rapamycin, what we see is when T cells are activated, a lot of these metabolites don't actually change dramatically. Lactate's here going up, pyruvate's going up. Alpha ketoglutarate levels go up, and they go up in sort of at least a largely TOR or rapamycin sensitive manner. Now, in the glutamine case, glutamine levels don't change after activation. Glutamate levels do increase, and this is partly rapamycin sensitive, not a change in oxaloacetate or aspartate here. So to suggest that maybe glutamine to glutamate, alpha ketoglutarate, like maybe this is an important pathway. And one way to test that is we made a knockout in glutaminase, but there was also a very specific inhibitor from Calathera called CB839. It blocks glutaminase 1. And then there's an inhibitor of the pyruvate carrier to get pyruvate into the TCA cycle. That's UK5099. So if you take T cells, stimulate them, and give these inhibitors, and just look for respiration, so what's contributing to the TCA cycle and mitochondrial metabolism, see that neither the pyruvate carrier nor the glutaminase inhibitor on their own do a whole lot. But you put the two together, and you start to get this decrease in respiration. So the T cells are using both glucose and glutamine for mitochondrial fuels, and there's some redundancies between the two. All right, so they're both going on. So that was good. So then we went and looked at the knockout mice. We mentioned we made a glutaminase knockout. This just shows you we have the knockout. It's a flox mouse. We crossed to a CD4 Cree. So that's the only Cree I'm going to talk about today. So in a resting state, you just look at these mice just off the shelf, they're fine. And actually, this is some flow plots, wild type, and then the knockout, again, just a CD4 Cre knockout. There's really nothing that stands out. It might be a little tiny bit of an increase in the Tregs, but it's not significant. So the resting cells don't need glutaminase. They don't use it, or it's not essential at least. Presumably because glucose can make it up. There's redundancy. Now, where you see the defect is when T cells are activated and have to do this switch to this anabolic program. So, of course, anabolism cells are going to grow. So ordinarily, if you look at forward scatter as an indicator of growth, wild-type cells start to grow. The knockout cells can grow a little bit, but they don't grow as well. And if we look at other activation markers, increase in CD44, decrease in CD62L, increase in CD25, proliferation with cell trace violet, the knockouts just don't do as well sort of across the board. So they don't grow as well, they don't activate as well, which is kind of exactly what you'd think. We knocked out a major fuel, and the cells don't do as well. So that's all well good. But where it got interesting was when we did a little bit, I'm going to show one more slide about in vivo, but then the functional aspects of this. Right. So this suggests that they're not actually activating so well, so there's something going on. But then we did this is actually with some help from Mark's group. We tried to see what do they proliferate in vivo, because that was all in vitro. And in vivo, I'll just put both these up, it's kind of a mixed bag. So this is immunization with 2W peptide. And actually, of course, there's very, very few cells that we measure ordinarily. After stimulation, there's a big expansion. So they can actually expand the knockouts in response to this strong stimulation here. In terms of homeostatic proliferation experiment, they have some defects. They don't homeostatically proliferate as well. So it seems like in vivo, there's some context specificity to needing to GLS1. Well, this is transferring T cells into a rag mouse, and we look like 5 days later, right? So it's kind of an early phase in that. So they're not as good here. So in a response to a strong stimulation, they seem like they can do it right, but not always. Right. All right. So that shows that they can actually activate okay. So what do they do functionally? And so this is where the data got really sort of surprising to us. And so this is if we're looking at T cells that are activated in vitro, and looking for production of cytokines, interferon gamma, IL-2 in this case. So T cells that are stimulated vehicle, now these are not they're given some IL-2, but not a whole lot. They're making IL-2, they're not making much interferon gamma. But the T cells that were treated with the glutaminase inhibitor, even though they're not activating by activation markers, they're not proliferating as well, they're making whopping amounts of interferon gamma here. So they're starting to differentiate down this fate. And so this is quantified here. And actually, if we look at TBET, transcription factor that can drive this, they actually upregulate TBET pretty strongly. Okay, so it kind of looks like we're maybe turning down the proliferation, but actually promoting a differentiation fate instead. Okay, so that made us go back. So this looks like the T cells are actually becoming sort of Th1-like by blocking glutaminase. So we'll go back to these data I showed you already and try to pull out some of the same glutamine metabolites. And so these are the things I already showed you, really, but just looking at naive Th1s, Th2s, Th17s, and Tregs. And we'll just zero in on glutamate here. So if you look at this, there's nothing that really stands out about Th1 cells in particular, but Th17s are interesting because they have a very low glutamine, very high levels of glutamate. So you can take the ratio of these signals, sort of a product to substrate sort of approach. So Th17s are very, very high. So it suggests that Th17s might be actually doing a lot of this metabolic pathway relative to the other subsets. Again, nothing really special about the Th1s here, but Th17s stand out. Right. So then we went back and just did the comparison between Th1s and Th17s to try to see how do they need these. So if we differentiate Th1s, Th17s with or without the inhibitor here and did the metabolomics, see the Th1s are affected. Their metabolism changes when you block glutaminolysis, but not dramatically. But the Th17s much more strikingly affected by inhibition. And if we look functionally at these cells, the Th1 cells, when you block, this is the knockout now, when we don't have glutaminase present, the Th1 cells can still proliferate okay. The Th17s are, again, much more strongly affected. A few cells are proliferating there, but a lot aren't. And if we look at viability, the Th17s live better for us sort of in general, but they have a decrease in viability in the knockout, whereas the Th1's not really affected. So it does seem like the Th17s and Th1s are very differently using or reliant on this pathway. Okay, so that's just the global levels, because we're interested in the metabolism aspect of this. Totally, glutamine and glucose metabolism should be linked. And so if you take the Th17 cells and look at glucose uptake after blocking glutaminolysis, that decreases. Okay, this makes sense. These pathways should be linked. But the Th1s are the opposite. The Th1 cells, when we block glutamine metabolism, their glucose uptake actually goes up. So they've uncoupled these pathways and seem like they're able to compensate and increase glucose as a consequence, where the Th17s can't. And if we measure glycolysis using this seahorse ECAR assay, it's the same story. Th1s increase when we block the pathway. Th17s more glycolytic to begin with, but they decrease. So they're opposite. So this would respond very differently. Okay, so that's the metabolism. What about just the differentiation here, based on some transcription factors, T-bet, R-gamma T, FOXP3. The knockout here versus the wild type. We stimulate and try to differentiate Th1s. The Th1s that we differentiate that are GLS knockout, they express more T-bet. It's harder to make Th17s. They express less R-gamma T. FOXP3 pretty much unaffected. Kind of seems like the Treg don't care about this enzyme. This enzyme's not really important for them. But this is quantified on the right. Th1s have more T-bet, Th17s less R-gamma T. Treg don't care. And that's the knockout. This is the bottom. This is with the inhibitor. It's the same story. Th1s do better when you block this pathway. Th17s less so, and Treg not really using this enzyme, or certainly not dependent on it. Okay, so that's kind of cool. It sort of suggests we really are bifurcating at a differentiation level here, potentially. So we can look functionally, and these are looking at cytokines now, wild-type cells to differentiate Th1s or Th17s or the knockouts. And we get more double interferon gamma IL-2 producers here when we differentiate Th1 cells if they're deficient for GLS, and fewer IL-17 producing cells, and quantified over here. And the exact same result if we use the inhibitor. So it's all very consistent. So mechanistically, we're still trying to work out why do they have these differences. We have a few ideas, but this is just an RNA-seq to try to get started at it. So Th1 cells treated with or without the inhibitor, Th17 likewise. And a few things to point out here. One is the Th1 cells. Again, they look like they're differentiating more. And if we go down this list here, there's a few things that say, you probably can't read it. TCF7 there is actually going down, so this would be consistent with greater differentiation. Signals like interferon gamma are actually going up. And there's a few other changes like DMNT1 here, so this could have some impact. Th17s is a little more of a mix. A lot more things in terms of things going up and down. But if you take the pathways for the Th1s and try to see what are the pathways that are really increasing. And so just do gene set enrichment analysis, E2F, G2M, mitotic signal. So cell cycle things are increasing. mTOR signaling, MYC, IL-2 signaling here, and glycolysis also going up. So it looks really like the Th1s are differentiating, proliferating, turning on anabolic pathways more so. Okay, so then we have to go back to this. I showed you glucose is important, so now glutamine is important, but it has this very specific Th17, Th1 sort of differential roles. So what does glutamine actually do? So glutamine, it's fuel for the TCA cycle, maintains redox, it's a nitrogen donor, but it also regulates epigenetics. And I kind of want to switch to this just for a little bit. So by supporting the TCA cycle, that gives you citrate. Citrate is exported from the mitochondria and provides cytosolic acetyl-CoA for all acetylation reactions. And then at the same time, alpha-ketoglutarate itself is used in the demethylation reactions. Okay, so it's actually it's sort of simplifying a few steps here. But of course, the methyl transferase will put a methyl on, but then demethylases actually use alpha-ketoglutarate as part of the reaction, and alpha-ketoglutarate is converted to succinate. Okay, so it's not just there; it's actually part of the reaction for demethylation. So as alpha-ketoglutarate goes down, demethylation decreases. So we have a lot of potential changes at the epigenetic level. Okay, and these metabolites, as they change, can have epigenetic implications. And so this is a slide that I've taken from Jason Locosauer. He provided me, where so he's been interested in metabolism and epigenetics. And if you take the KMs and the substrate concentrations of different enzymes, kinases, for example, versus some epigenetic modifiers, what you see is that the levels of ATP as they change within a cell never drop to a point where they're ever limiting for a kinase. But if you go to the methyl transferases, the histone acetyltransferases, the metabolites, the substrates for these reactions actually do drop to levels where they can become limiting for some of these enzymes if you calculate the KMs. All right? So the metabolic levels can actually have pretty potent epigenetic effects. Right. So then we went and looked to see, well, if alpha-ketoglutarate, we think the player here, what really changes? So Th1 cells do have a decrease in alpha-ketoglutarate when we block glutaminase. The Th17 cells, it doesn't actually change. And the reason we think it's not changing is because we think they're shuttling a lot of things to alpha-ketoglutarate, like other pathways that can generate alpha-ketoglutarate are depleted. So we think Th17s are just trying hard to maintain this, but the Th1s don't. So that goes down. So this decrease in alpha-ketoglutarate might be contributing to the phenotype. And so we went back and did an experiment where we just tried to replace the alpha-ketoglutarate. So here what we did is take vehicle CB839 or CB839 plus dimethyl alpha-ketoglutarate, or cell permeable alpha-ketoglutarate. What you see is the glutaminase inhibitor, we get increased cytokine secretion, but you put the alpha-ketoglutarate on there, it takes it back down, restores it back to what it was. Now I should point out this doesn't do anything at all to the Th17s. So the Th17s are not rescued by alpha-ketoglutarate. They seem like they have some different mechanism. Okay, we think actually it might have something to do with the redox balance more so than alpha-ketoglutarate. But nonetheless, this shows that the alpha-ketoglutarate is contributing to the Th1 phenotype. All right, so if alpha-ketoglutarate TCA fuel, but also regulates epigenetics, one of the things to do is just look at the global sort of epigenome of methylation here. This is H3K4 trimethyl or HK27 trimethyl, normalized to the total, treated with or without the inhibitor. We see Th1s have a decreased trimethyl, Th17s it actually goes up. So they're responding differently to this change at a global level, at least. Now, because in principle we have any methylation change could be changing, any acetylation could also be changing due to the TCA and citrate levels. Rather than go through and do a whole series of ChIP-seq, we did an ATAC-seq experiment instead. So what ATAC-seq does is it's a transposon-based system where you insert an oligo into open chromatin, and you just sequence through. So you can just identify open chromatin, basically. And so when we do that, just try to see how does the chromatin accessibility change rather than focus on an individual mark. Which sees in Th1 cells, there's more genes that have sites that are opening than in the Th17 cells where it's more genes that have sites that are closing. So again, from a chromatin accessibility, they're going opposite. And if you compare these sites, there's some overlap here, but there's a lot of independence as well. Okay. So you can go through all these sites and look at the different tracks. I'll just show you one of them. This is interferon gamma. Of course, should be opening with the Th1s, and we get a nice shoulder on this peak in the promoter region where it's starting to open, and there's a few sites within the gene that are becoming more accessible as well when they're drug treated or glutaminase inhibited, and that correlates with an increase in the interferon gamma expression. Okay. So there are some epigenetic chromatin accessibility changes here that are correlating with the effect. Right. And if you can go through these peaks and try to pick them out and see where are the consensus sites, and it's kind of messy because there's a lot of things that change. But if you just sort of go into these big transcription factors, things that are more open in Th1s and Th17s and less open, and the thing that's really worth pointing out here is the ones in red. It's the same four families of transcription factors that really show up across the board, AP1, ETS, IRF, and CTCF, that are changing. And the CTCF is really the really worth pointing out because it has been a transcription factor that has been shown to respond to changes in glutamine. So it's been known to respond to alpha-ketoglutarate. So it's actually really nice that showed up. It just sort of showed that we are getting changes consistent with that. And of course, these others have lots of roles immunologically as well, which is great. Okay, so that's all well and good, but how do we actually get to that point? And increased glycolysis and these epigenetic changes downstream of alpha-ketoglutarate. How does that actually cause the functional change? Well, one of the things we've seen is, of course, glycolysis is up. And one of the drivers of glycolysis is in the PI3 kinase TOR signaling pathway. So if we look at phospho-S6 as an indicator of AKT and mTOR signaling, Th1 cells, when they're treated with the inhibitor, glutaminase, their TOR signaling actually increases, whereas Th17s, it goes down. Of course, this can drive a lot of glycolysis. Now, there has to be a signal to activate this in the first place, so IL-2 is present, and this might be doing it. So that sort of gave us the hypothesis that we have a series of gene expression and chromatin changes, and maybe what the things that they're doing is, in essence, sensitizing the cell to IL-2. So we get a stronger AKT mTORC1 signal. This then increases glycolysis and effector function. So this is sort of the model that we went with. And so then we went back and tested, well, if that's the case, then is the IL-2 really the key here? So normally this is condition we'd use if we give IL-2. The vehicle-treated cells have a certain amount of interferon gamma, IL-2 double producers. That's increased with the drug treatment. If you don't give anybody IL-2, nobody makes a lot of cytokine. And in fact, the drug doesn't work anymore. You don't get that boost. And if anything, the cells are doing worse. Okay, so this increase in cytokine secretion is dependent on having that extra IL-2 there. Now, so we can go back and then say, is the TOR pathway important? And this is a little less clear, but it still makes the point. So if you give IL-2 and rapamycin, giving the rapamycin sort of after 3 days of stimulation, it doesn't actually do anything to the cytokine secretion, which is a little surprising, but if you think about it, so rapamycin is actually quite a good immunosuppressant, but where it's really strongest is in that initial activation, that exit from quiescence. When cells are activated, it's not anywhere near as potent, so nothing really what happens. But it does still have an effect on the inhibitor-treated cells. So we do start to bring that cytokine secretion. So that boost in cytokine is dependent, at least partially, on signaling through mTORC1. Okay. So this gives us this model. So glucose is important. Glutamine is a fuel here, but it also affects chromatin changes and something, we don't really have an idea exactly what's going on, but the Th17s require this. Chromatin modifications, we think this is affecting the sensitivity of IL-2 and mTORC1, and then this affects the differentiation of Th1s, Th2s. So that's about as far as we are with the model. We have a few candidates in there, but that's the bulk of it. Okay, so that's all great, and that's all built on pretty much in vitro studies. And of course, metabolism in vitro and in vivo are very different. So then the question is, what does this do in vivo? Does GLS regulate T cells in vivo? Do the Th17 cells work poorly if you block GLS, which would be a prediction? And do the Th1s and CTLs work better? Now, I haven't mentioned anything about CTLs here, but I should say that they look pretty much exactly like the Th1s. If you block glutaminase on a CD8 T cell, it will express more granzyme B. Okay. So do they have these different functions? And this is where we started to work with people like Bruce, who helped us on the Th17 question, and then we worked with a couple other different groups on the Th1s and CTLs. So this is what Bruce's group. So this is, as I say, one of his chronic graft versus host models that has a Th17-mediated dependence for certainly the lung inflammation we're showing here. So this is transfer of bone marrow alone, so there's no T cells, there's no allo response, so the lungs are clean. If you transfer in the wild-type T cells that have this allo response, histopathologically, you can start to see some infiltrate here, and this is scored the inflammation down below, the histopathology score. But if you use the knockout T cells, glutaminase knockout T cells, the lungs are pretty much normal. It might be a little bit going on, but very little, and the pathology score is normal. And if we look at the cytokine secretion, interferon gamma typically goes up. This is a non-significant change, but it does look like there's a trend down here with the knockout. But the knockouts are definitely making less IL-17. Okay, so this Th17-mediated inflammation here definitely seems to require glutaminase. So that's good. So that means the Th17s do need glutaminase in vivo. All right, so what about the Th1s and the CTLs? So here we switched to some cancer models. This is actually a CAR T cell model, so a chimeric antigen receptor model. So we take wild-type or knockout T cells, purify them from these various mice, and then infect them with a CAR construct. So it's a specific for CD19, and either lacks a cytoplasmic tail, that's our delta Z control, or has a CD28, CD3 cytoplasmic tail. And then we put them into mice and basically see what they can do. See, can they eliminate B cell targets, for example. Right. So this is a mixture of CD4s and CD8s in this. We don't really distinguish between the two because typically when they do these CAR therapies, it's just bulk T cells. Right. All right, so these are the data. So this is using the wild type and the knockout, either with the delta or the CD3 zeta, CD28 tail. And what you can see, and those are B cell targets. What you see is that day 14, after transfer, the wild type CAR T cells are able to eliminate B cells. Then so can the knockouts, right? There's a little gap there, but that's not significant. Day 28, though, the wild-type guys are, for the most part, there's some starting to lose it, but they're doing okay. But the knockouts can't function anymore for the most part. So the knockouts have this transient ability to work, but they don't seem to be able to sustain it if they just have no glutaminase whatsoever. So this was a little disappointing, but it really suggests that either, again, the cells are differentiating, so either there could be a terminal differentiation, or alternatively, there could be you just need glutamine to some level, and our acute in vitro assays weren't picking it up. And that's a question we're working on right now. We don't really have a good answer for it just yet. All right, but they could work initially. But then we had this sort of other option. Because we did have these changes in the chromatin accessibility, and presumably sort of an epigenetic sort of aspect to it, we could go back and do the experiment differently. So here we took wild-type T cells and stimulated them and plus and minus the inhibitor, just for a transient period of time. The idea being that if we are modifying the epigenome, the chromatin accessibility, that could be a lasting effect. So then we could go into mice where there is no drug, there's no inhibition, and try to see what happens. If we test specificity to a virus in a viral infection model, or go back to the CAR T-cell model and try to look at how efficiently can they eliminate the B cells and how long do the cells live. All right, so these are the data. This is both in vaccinia and a CAR T-cell model. So this overall result is they seem to be better. Okay, so if we treat the T-cells, do this transient treatment, and then if they're specific for vaccinia here, and this was done with Roger Kishton and Nick Restifo's group, in both cases we get a good elimination of the virus. That's fine, no difference there. But the big difference is even this is a couple months later, we have an increase in the number of virus-specific cells that are present. This is an earlier time point, but you look out 2 months, and it's about the same difference. So we have accumulated more memory cells as a consequence of this initial reprogramming. And it's the same thing with the CAR T cells. They are eliminating the targets pretty much the same. We can't really tell any difference there. But one of the big drawbacks, one of the challenges in these adoptive cell therapy experiments is it's sort of a race between eliminating the target and losing the effector T cells, because the effector T cells are dying over the course of this process. But what we see here is the CAR T cells, if they have the delta Z with or without the inhibitor here, there's more of them present that were treated with the inhibitor. So blocking glutaminase in that in vitro time, transferring the same number of cells into the mice. At the end of the experiment, there's more of them left. Okay, so from a therapeutic perspective, this should be good, and presumably these would be better able to handle a tumor over a longer period of time. And we're moving into those levels of experiments right now. Okay, so this is the overall model. I showed you this model earlier. These are the conclusions. So T cells use both glucose and glutamine, and there's some redundancy, but the different subsets are different, and they each handle it in their different ways. With T cells themselves, of course, they need glucose metabolism for effector function and in inflammatory settings, and of course it's altered in tumor in TILs. But glutamine has this sort of interesting role that we're really sort of fascinated with, because it gives you both this energetic TCA source, but it also has this regulation of differentiation. We think it's downstream of its impact on the epigenetic sort of aspects of this. And whereas chromatin are more open in Th1 cells and closed in Th17, blocking GLS inhibition, and it does seem to have this effect where the Th17s need this pathway, and Th1s are actually repressed by this same pathway. And so I think there's a lot of opportunity now to try to see, well, what happens when we really start to modulate this in terms of functional outputs. Because the inhibitor I've talked about, the CB839, that's been developed entirely to target cancer metabolism. Okay, so it hasn't been used in any inflammatory settings. So right now they're in the mode of trying to move this over. Of course, every cancer company is trying to partner their products with PD-1 and CTLA-4. So they're doing all those clinical trials right now. It actually looks pretty good. Right? Some of the data is coming along. So there might be some opportunities where you can both hurt the tumor and help the T cells at the same time with the same drug. Okay. All right. So that's all I want to say. And then this is the group. My lab is partnered with my wife's lab. So Kim Rathmell there. That's why we're Rathmell squared. So she's also at Vanderbilt and is a urologic oncologist. And so I say we do kidney cancer because kidney tumors have the most CD8 cells, and that's super convenient. But the reason we do kidney cancer is because that's what my wife does. So she studies kidney cancer and brings all these tumors into the lab that we can then study. So this is our group together. And so the tumor work was all done by a previous postdoc, Peter Siska, and a fellow in the lab, Katie Beckerman, there. And the glutaminase work was all done, at least within our group, by Mark Johnson. Of course, we've got a lot of collaborators, a number of them here at Minnesota. And I am happy to take any questions. Thank you for your attention. Yeah, Amita.
Okay, wait for the microphone. We have 3 other locations that want to hear your question.
So the Th1 cells, I mean, I know you talked about them adapting very nicely to the absence of glucose and switching, glutamine and switching. Have you looked at fatty acid oxidation at all? Do they use it at all? Because it's interesting that their mitochondria are completely healthy. You know?
Yeah, no, we haven't actually looked at that. I mean, presumably they are doing a fair bit of fatty acid oxidation. Be another way to feed acetyl-CoA into the cells.
Yeah, right.
So we haven't looked at the pathways. We have kind of looked at a little bit, because fatty acids are not necessarily a great anaplerotic source to keep the TCA going, because it provides acetyl-CoA, but you still need the oxaloacetate to condense with that. So you still have to have something to get get more carbon into the pathway. So glutamine's not doing it. Glucose can through pyruvate carboxylase. Yeah. So actually, there's an increase in pyruvate carboxylase in those cells. So I think that they're using glucose glycolytically, but then also using it more in the TCA cycle as well. But fatty acids, we haven't. We've decided to set that aside for the time being. Yeah, Mike.
So I've got a question about the last part where you're looking at the effects of GLS inhibition in the CAR T cells. And so essentially, if you knock it out completely, you were showing that you couldn't sustain tumor cell killing. But if you did this sort of transient inhibition with the drug, now you're getting, like what you were predicting, better effector responses. I'm wondering, is a drug potentially doing something different than just a knockout? I mean, if you give the drug long term, do you mimic what happens with the GLS knockout? Or are there reasons technically that you can't do that?
No, that's a great question. The drug is probably going to be different from the knockout for a couple of reasons. We haven't done the long-term treatments in our experiment. Of course, patients are on this drug for a long time. And of course, whenever this drug has to be given twice a day, and so there's this up and down. You hit the trough, right? And so they typically get about 90% coverage with their drug when they hit the trough. So there's activity when the drug is given. So the knockout is quite a bit more severe than the. So even if there's that's an entirely on-target effect. There can be off-target effects or secondary effects that we're not getting. So it's not exactly the same. So we're wanting to move in to do these longer-term drug treatments and see if we get this, if it looks like the knockout. And transient things. That's the other thing. The clinical trials with this drug are all given where they put people on the drug and they just stay on it. Where our data would say if the drug actually is getting a sufficient coverage of the target, having a patient on it continuously may not be so good. You might want to instead have it on for a window of time, either scheduling effects or timing of the treatments. But we don't really know yet.
Is there any effect of the drug actually on the tumors potentially themselves?
Oh, yeah. So again, the drug
is a good thing?
Yeah. The drug was made because cancer cells like to use glutamine. And so it was meant to target cancer metabolism in this anaplerotic sort of aspect of glutamine feeding in to support the TCA cycle. That's why it was made. Some tumors are more sensitive than others to that. Actually, turns out kidney cancer is one of the more sensitive ones, just sort of by coincidence. So it kind of works here and there. It's a little spotty. But it definitely has, and part of the idea there also is that not only are you blocking the tumor metabolism directly, but you're changing, because the tumor, those cells may not die when you block the pathway, but their glycolysis goes down, so then the whole microenvironment potentially improves for the T cells. So in a solid tumor setting, you might be getting this thing where you're having these differentiation effects on the Th1s, hurting the tumor, but then also improving the tumor microenvironment. So there's a lot of different things that could be positive.
So fascinating work. It's really cool seminar. I have a question that's a little bit chicken and egg. So when you have these different T cell phenotypes that we're creating, mostly in vitro, do you know if when cells go to Th1 or Th17, is there an intrinsic difference in the expansion rate that maybe you're just exaggerating? Or is it really that they're wholly dependent on these different things? In other words, are they sort of adapted to do this, or are you forcing a requirement that then creates a change?
Yeah, it's a great question. I mean, there really isn't a solid answer for that. I mean, one of the ways to look at it is, it's something that we've been thinking about for a long time, is we see these different metabolic profiles in different T cell subsets, but they don't proliferate necessarily at the same rate. That's just at a very high level difference. And of course, proliferation rates are going to affect all these anabolic pathways. So what we've seen is that some metabolites are a lot of this just falls away into proliferation rate. But there's a lot of things that are not dependent on that and seem very specific for the subset. Now, whether they are driving it as cause or whether they're consequence, that you cannot know until you actually go into knock it out or target it in vivo and try to see what really happens. So, again, this is one of our big goals, is to try to get a better idea of these metabolic networks so that we can go in and target them and try to see, well, okay, if we block this specific pathway, are these cells specifically affected as we think they will be? But a lot of it can be proliferation, and so you don't really know till you try it. So. That's not a great answer, but that's kind of that's sort of where we are.
So getting back to Mike's question, is the inhibitor specific for GLS1 or GLS1 and GLS2?
It's GLS1 selective. So there's a GLS and there's a GLS2. And the inhibitor doesn't hit GLS2. Now T cells, different tissues are different, right? But T cells have much more GLS1 than GLS2. When they're activated, GLS2 increases, and so the fold change of GLS2 is actually pretty significant. But they're starting at a very low level. So it's about 20-fold more GLS1 than GLS2. But the inhibitor is still very GLS1 selective. So we think this is why we think, pretty much when we use the inhibitor and compare it to the knockout, the phenotypes, they're not always exactly the same, but they're pretty close to the same. We think that's because T cells are pretty dependent on GLS1. Now we don't see any effect of the inhibitor on Tregs, which is a little different from a few other papers that have targeted glutamine at different steps, where they actually have seen an effect on Tregs. And what they find is there's a boost in Tregs. So either that means that Tregs aren't actually doing glutaminolysis, at least through glutaminase, or alternatively, there might be enough GLS2 there that they're okay with that. But for the most part, the GLS1 is selective.
In a tumor microenvironment, there should be a lot of lactate because of the aerobic glycolysis in tumor cells. So I'm wondering whether the lactate can be used by T cells to rewind cellular metabolism, or there's any signaling effects on T cell function.
Yeah, so we haven't looked at that ourselves. A few other groups have. Marina Kreutz at Regensburg in Germany had a really nice story in Cell Metabolism, I guess about a year or so ago, where they showed that the lactic acid and lactate in a tumor can selectively inhibit effector T cells, but actually Tregs like it. Treg will take up the lactate and burn the lactate, and it's quite a efficient fuel for them. So the lactate is used by the T cells, but again in a substrate-specific manner that seems to inhibit the effectors and promote the Treg.
Other questions or comments?
What happens with malnutrition in terms of T cell immunity? Do your results tell us anything about that?
So I had a fellow in my lab who was working on this sort of question, and I can tell you her data. So, of course, obesity drives inflammation. Malnutrition is associated with susceptibility to infection. Right? This has been known for a long time. Interestingly, that's not the case in anorexics. Anorexics are not more sensitive to infection, so there's actually it's not as simple as just malnutrition. The extra factor in the anorexic cases, they tend to, that's almost always associated with an exercise compulsion. So exercise might modulate it. So it's not so simple. But what my fellow did was she took animals and would fast them and then look at the immune responses downstream. And the T cells start to turn off. Tregs are favored. Th17s don't do as well. Th1s don't do as well. The sort of twist that she had that I think is kind of neat, so this is Nancy McIver, is a lot of hormones change in fasting case. Like leptin in particular goes down, and leptin is known to be an inflammatory cytokine, of course, made by adipocytes. So if she took the mice and fasted them and injected them with leptin at the same time, then she didn't see any of those changes. Right? Then the T cells were fine. So it wasn't just this nutrient change, it was a hormonal change downstream of something in the adipocytes that then the immune cells were responding to. So anyway, she also sees that insulin and IGF can be pretty potent regulators of immune function too.
Others?
Nancy McIver.
So is there any relation between the TIL efficiency into the tumor and the glucose metabolism?
Well, the glucose metabolism goes down in the TILs, and it starts to go back up, I guess, if you can re-stimulate them. So I guess there's an association, but I don't know how linear that actually is. All right, well, thank you very much.
Thank you.